Hello, welcome to the next portion of the class. So we're going to go through a few different topics here. We're going to start off talking a little bit about the history of neuroscience. Very interesting, sort of helps us to understand why we are where we are. Um, in the history of this field and where we're going. Best way to know where we are and where we're going is to know where we've been. So we're going to do is spend a little bit of time on this. And then we're going to move to introduction to the nervous system, where we're going to explore some important concepts related to the structure and function of the nervous system in general. And we're going to be emphasizing the central nervous system. And then we're going to talk a bit about neuroanatomy. So the first part is the brief history of neuroscience, and I call it brief history because this is sort of a small portion, okay, of all of the major discoveries that have led up to our current understanding of the nervous system and the brain. So, first of all, one of the things that I really enjoy about neuroscience and the cool things about neuroscience is that this field really is a marriage of several different types of science. So, neuroscience encompasses fields including molecular biology, which really deal with structures and molecules and at a very specific level, cellular biology, and pharmacology, and chemistry, organic chemistry, etc. So, neuroscience is a very large area and there's a lot into it, okay? But essentially, what we're dealing with here is an interdisciplinary field, okay, that tries to study and investigate and understand the structure and function, okay, structure and function of the nervous system in the brain. And in fact, when we get to neuroanatomy and some other topics, we're going to see that the relationship between the structure and the function is very important. So oftentimes when you study anatomy, we study structure function types of relationships. Okay, so this picture here on the right side, these are actually neurons, okay, that are forming lines or synapses together here that are actually going to be communicating with each other. And we're going to spend some time going through the details of that later. So how did it begin? Well, the history of neuroscience, like I said, is sort of a fascinating one. So we can go all the way back to about 7,000 years ago, okay? And from fossil records and discoveries from, the types of people who dig down into the ground and find bones and things like this, knew that the ancients knew a bit about brain function. And essentially, really what they knew was that the brain controlled important functions of the body. And now nobody really knew for sure exactly how this was or exactly what it was. But what is interesting about the history is that by studying the skulls and the remains, okay, of previous, old human ancestors, what was discovered was a lot of sort of holes that look like this, okay? Now, the practice that was practiced back then was called trepanation, okay? So, they would trepan or trephan, depending on where you're from and how you pronounce it, which basically means burrowing a hole in the skull, okay? Now, you may think to yourself, yeah, they were probably fighting each other and things like this, maybe struggling for control over land, and this is how these holes entered into these skulls, right? So scientists began to discover these, but what they found actually was something interesting, was that around the region of these holes that they were finding in the skulls, okay, there was actually evidence of new bone formation, okay? And so what this told scientists was that this new bone formation is actually some evidence that these holes were not made in attempts to kill people like you might have originally thought, but actually maybe as an attempt to heal people, right? So this is the first sort of clues that we have from history that people knew that there was a definite correlation between the function and what was inside your head, okay, i.e. your brain function. Okay, so these might have been some of the early attempts that people had used to actually try to cure people who might have had some kind of disorder, disability, or something like this. Okay, we can look back at ancient Greece, okay? And the Greeks really were famous for many things, but one of the things that they determined was that the brain is actually the seat, okay, of sensation, right? So when we say sensation, we're talking about the ability to perceive things from the environment, okay? And there were a couple of important people. Hippocrates, all right, is credited as one of the first people to actually decide and profess that the brain was actually the center for intelligence, okay, the seat of intellect in the body. Whereas Aristotle still contended and fought that the heart is actually the center of intelligence. Now, many people thought this back in this time, okay, but it was Hippocrates who actually ended up being right in the end. And in fact, Hippocrates' legacy still lives on. A lot of you may know that as a medical student, when you finally finish and you're being awarded your medical degree, you will be asked to recite something called the Hippocratic Oath, okay? And that's based on an original form of it. Now, it's not exactly the same anymore, and there's actually several different versions of it, but Hippocrates, okay, was the first person we think who came up with a version of the Hippocratic Oath, and that's why it's named after him, right? So he was a physician back then, and so this is sort of where that came from. Interesting. And there's a little picture of our Hippocrates. So if we move forward, we get to the Roman Empire, okay? Again, Hippocratic view of brain functions. So the idea of Hippocrates lives on, okay? And we see that there is another key individual in history by the name of Galen. Galen was a very interesting fellow because in addition to being a physician, he was actually a doctor for gladiators, all right? And so what Galen would do is he would have the fun, and I'm sure quite challenging task of actually trying to put these gladiators back together after their battles, okay? So this is kind of a little image here that depicts Galen standing over the body of a gladiator who has obviously been in some sort of very difficult and probably gruesome fight to some extent, okay? In addition to that, Galen also was an avid sheep dissector, very interested in the structure and the function of the human body, okay? And so he would use what was available to him to try to learn, and one of these things was sheep, okay? One of the first things that he determined when Galen first bored through, okay, into the brain was that there were actually portions of the brain that were hollow. Okay, and these hollow portions of the brain are really what we later found to be ventricles, right? Okay, and when he saw these ventricles in here, okay, that essentially allow fluid, right, to pass through the brain, he determined that so Galen determined that this idea of these ventricles and these hollow spaces where fluid could pass around through the brain was consistent with a popular idea at the time. And this idea was that there was actually a natural balance within the human body of four humors, okay, or sort of liquid, right, aqueous substances that were necessary for life to sustain, okay? So again, this was a finding that supported the idea at the time, even though this idea at the time of the four humors actually was wrong, right? But what Galen found, and this sort of supported that, so people continued to think this, okay? It's kind of an interesting thing, actually. Then we're going to move to the Renaissance era, okay? So from the time of the Renaissance all the way through about the 18th century, there was another key individual by the name of Vesalius. Vesalius really took human anatomy to another level, okay, through many dissections that he performed and many very detailed drawings of the human body. So if you ever have a chance and you're interested, go ahead and take a look at a book, okay, or try to get it online and find some images, okay, of Vesalius's drawings. Now, a lot of them are sort of entertaining because he depicts bodies in very strange and somewhat compromising positions, okay? So we can see sort of one here. This is some kind of looks like something you might see on Halloween, right? Put it in somebody's window. But he did learn quite a bit about the structure of the human body and how it was all assembled, okay? Also, he discovered some other things, and some other things were during this time period, okay, were discovered that were very, very important, okay? For example, this period of the Renaissance through the 18th century was actually when we found that there was actually white matter and gray matter, okay? So we're going to talk about the white matter, okay, is really the axons, and those are the portions of the neurons that actually relay the information, okay? So we have these signals that travel from the cell body to the terminal, and they travel down axons, right? So this white matter, okay, which relays information, right? And really what happens is it goes to and from the brain. If we're talking about peripheral and central nervous system differences, and we'll talk about that later, okay? And the gray matter of the brain, which is that in the spinal cord as well, okay? So even in the brain and the spinal cord, we have white and gray matter. The gray matter is actually the cell bodies themselves of neurons. The distinction between the central nervous system and the peripheral nervous system was also determined during this time, and through close examinations of brains, it was discovered that there are actually bumps and grooves, okay? Now we call these very particularly gyri in the case of bumps. So a single one is called a gyrus. You can see there's a gyrus shown here in this image. It's just this bump on the surface of the brain. And grooves called fissures and sulci, okay? So here is a fissure. So you can see this is actually a groove in between the left and right cerebral hemispheres, okay, straight down the middle. And this is a fissure. And then we also have smaller ones here, okay, smaller grooves, which we call sulci. So a single groove, which is shown here, is a sulcus. Okay, sulci would be the plural for that. All right. Now, what was determined was that if you looked at the brains of various different individuals and you studied the pattern of the bumps and the grooves, right, the gyri, the fissures, the sulci on the brain, you would find that they were present in a very remarkably similar pattern. Okay. And what this actually did was it showed us two different things. Number one, it told us that there were actually points in the brain that we could use as landmarks, okay, such as this central fissure, okay? And through using these particular bumps and grooves that were very prominent, it kind of helps us to navigate our way around the brain, and we can start to talk about different parts of the brain, different regions of the brain. And the reason why that's important is because people started to think, well, we have this very consistent pattern of bumps and grooves in the brain, and probably these lobes, okay, these different regions in the brain have very specific functions. And this was really the beginning of localization of function, as we would call it, okay, in the brain. So here, this is just kind of another close-up view where you can see a gyrus, okay? So many of these would be gyri, okay, but this actually one is actually just the gyrus, okay? And the sulci here are in the middle, so this is a sulcus. So you can kind of see these here in some better closer up view compared to this image here, which is a little bit further away, right? 19th century, huge explosion, okay, in information. So we learned a whole bunch of things, and there were some key experiments about this. So first of all, the idea of nerves as wires was validated experimentally, okay, and this was really the very first specific glimpse into the ways that nerves actually conduct action potentials. Okay, so we're going to go all through the function of the neuron, but essentially you may or may not know that the neuron acts by firing what we call an action potential, and there's actually an electrical current that travels down the axon, okay, from the cell body to the axon, down the axon to the terminal, and it then activates the next cell, which does the same thing, okay? Very similar to the way in which a wire conducts electricity. And so this idea was hypothesized, and it was actually confirmed experimentally, okay, by what we call transection experiments. So a transection is essentially a cut through, okay? So by transecting a nerve, so in other words, severing a nerve, cutting it, we could see that there was function lost, okay? So it was known that if we have a nerve that runs, okay, let's say from an area in your spinal cord to your right, one of your appendages, let's say it's your right leg, okay, and we cut that nerve and you then lose function of your right leg, we can presume, okay? Or we have evidence that that nerve is actually carrying information that you need to move your leg, okay? So nerves as wires. The other part of this was actually measuring, okay, the ability to move muscles in response to electrical stimulation, okay? So this was where electrodes are hooked up, okay? And in these experiments made by Galvani and his colleagues, stimulating electrically, okay, one particular place is actually going to cause a movement in the muscle, all right? And so what they saw was that if you take the nerve, you apply a weak electrical current, you can actually get a twitching in the muscle. So these two types of experiments together really reinforce the idea of this nerves as wires, okay? And then even more information about brain specifically was determined by Florenz through experimental ablation. And this method is one that is still used today, currently. So in experimental ablation, essentially what we do is we cut a piece out and we observe how the function changes, okay? Very basic type of experiment. We determine whether we can get loss of function, okay? So through these types of experiments, by ablating, okay, or removing certain parts of the brain, we can look to see what's going to happen with the movement. And indeed, a couple of key findings from these experiments were, number one, that the cerebellum is actually involved in the coordination of movement, okay, pretty important, and that the cerebrum, okay, or what we normally think as a lot of times as the cortical area, okay, the cortex, is involved in sensation and perception, okay, so being able to touch, see, taste, okay, perceive all of the different stimuli in your environment is really important to have an intact cerebral cortex, okay, and some other structures in the cerebrum as well, okay. So here you can actually see sort of this is a muscle fiber being shown here in what we call myofibrils. So within each one of these, there's actually it's driven by ATP really, but we actually have this is how skeletal muscle is contracted in the body, okay. And here we have a neuron that's actually forming what we call a synapse, okay, onto this muscle. And by stimulating this motor neuron, we can actually cause this muscle to contract, and that's the twitch, right? The twitch that you feel. So that was the first sort of experiments, okay? The next part was really something called phrenology, and this was Gall, okay? Now, phrenology was interesting, to say the least. It wasn't exactly right, but it wasn't entirely off base, okay? So the idea with phrenology came as a consequence of us starting to sort of figure out that there was this localization of function in brain. And what Gall noted was that since we know that certain parts of the brain might have certain functions, and we know that people have different shaped heads, okay, maybe the shape of the skull or the shape of the head is a consequence of the underlying brain. And so for this reason, can we actually study the bumps on the heads of an individual and be able to tell, for example, if they're going to be a good sculptor, if they can paint? What the likelihood is that they can solve a very difficult math equation, okay? So Gall's idea here was that we might actually be able to map all of these regions, and simply by feeling the surface of somebody's head, okay, which reflects the skull underneath, right, and therefore the brain underneath that, we can feel the bumps, and based on these different bumps, we can tell whether what type of profession somebody might engage in, right, or what somebody's good at, what their talents are, what their talents aren't. Not an exact science, okay? It wasn't really correct, but it was neat, and it was something kind of interesting, all right? But what was correct was actually the speech center of the brain, and this was an important one that was actually figured out as well, okay? So here on this image of the brain, you can see this little region here, and this is called Broca's area, okay? Broca's area. And this was an important discovery because we found that there's actually very specific parts that regulate things like speech, okay? So with Broca, it's very difficult to speak, okay? Even though you might be able to understand something, but actually being able to form the words is not possible, okay? And there's actually another speech area that's sort of the opposite of that, which is called Wernicke's, and that's back here. Okay, but these discoveries were important monumental discoveries that led to, from the very beginning, where simply it appeared that people were drilling holes in skulls because they figured that the brain controlled people's function, and maybe somebody had some sort of disease or disorder, and drilling a hole maybe would let out some of those bad things that were infecting the brain, right? I mean, that's really what they were doing, okay, with trephination. Um, all the way up to the point where we're actually conducting controlled experiments, okay, with things like nerve stimulation and experimental ablation to actually confirm that indeed there are certain parts in the brain that do regulate specific functions, okay? And now, really, what we do as neuroscientists is that we use a variety of different approaches, okay? One of my favorites here, caffeine, right? Molecular neuroscience and cellular neurosciences are fields that really go hand in hand, okay? But what we see is this idea of reductionism, okay? So we can take a problem, okay, whether we're trying to explain a behavior or we're trying to explain a disease state or even a normal physiological function for that matter, okay, or cure a disease state, and we can look at it at many different levels of analysis, okay? Now, to understand exactly which molecules and which individual proteins and which individual transmitters are responsible for something, you're really looking at the molecular level, okay? Or maybe the cellular level, okay? What cell types are responsible for immune function, right? Immune cells, there's lots of different kinds. Maybe there's a certain particular type of T cell that you're interested in. Okay, so that would be more of like your cellular neuroscience. Okay, this is a really nice staining of some different neurons, okay, in the brain that actually, you can see the cell bodies here, and these are the axon areas coming off, and you have all these dendrites branching, okay. But then we can actually move forward into a systems approach. So we go from the individual molecules and the proteins involved in something to how they all work together on a cellular level, okay, to how the cells actually connect in synapse, like shown here, okay, into a systems approach. And so we're going to be looking at some various systems in the brain, okay, dopaminergic system, serotonergic system, okay. These are systems that work together to actually regulate lots of important functions, all right. Once we know those kinds of functions, we can actually look at things at an even larger level, which is behavioral, okay. So for example, addiction, very interesting field. What is it that actually causes somebody to become addicted to a drug? Can we trace this back to actual individual molecules? Okay, if we know something about certain molecules that are released, okay, or that are signaled when somebody takes a drug that's addictive, can we actually use this information to change their propensity to become addicted? Okay, that would be a behavioral. Can we change the behavior? Okay, and then we can go all the way back up to cognitive, which really is a huge term because it means so many different things. There's so many different aspects of cognitive neuroscience that it just sort of depends on what you're talking about. Okay, but ultimately, what we are sort of taking home from this, if anything, is that the brain is extraordinarily complex. Okay, it does work on a systems level. Okay, understanding the brain at a systems level probably is necessary to be able to really be effective in behavioral or cognitive neuroscience, because you really have to understand how everything works together. And in this class, we're going to do some of that, and we're going to explore some of these systems individually. We're going to be exploring certain cell types, and we're going to get into some of the cellular and molecular aspects of how cells function together in the central nervous system, and particularly in the brain. Okay. So how have people actually been able to discover lots of what we know now? Now, we did mention the sheep dissections earlier, right? So animal molecule models, rather, are very important in research, and it's very important, has been very important in the study of neuroscience, okay? So what we can do is we can actually model, okay, human conditions. We can model normal physiology, okay, in animals that have physiology that are similar to humans, and we're going to talk about some of those. And we can also model pathological conditions, okay? So animal models allow us to sort of recreate things that we see in humans to try to study and understand them, okay? And the reason why this works is that depending on the species you're talking about, okay, and the specific system, right, the specific body system you are investigating, Okay, there is likely a species that has a lot of similarities. Okay, and one of the ones that is studied a lot in neurodegenerative disease tends to be pigs, rats, mice. Okay, things like that. So by using these animal models, we can actually understand very, very important aspects of brain function. And the reason that we want to understand these aspects of brain function is because if we understand how the brain works, then we can figure out how to fix it when there's something wrong. Okay, so for example, in disease pathology, right? The other obvious advantage to using an animal model, okay, is that it's much cheaper than doing a human study, and we have a lot less ethical issues to deal with. Okay, so you can imagine, let's say, for example, we're trying to treat something like Parkinson's disease, and there is a new drug that has been created, and we think that it's going to be great, and we think it's going to be the next big thing, and it's going to help a lot of people who have Parkinson's disease. Well, if, for example, we have 10 patients with Parkinson's disease, okay, we want to be able to tell whether or not this drug is going to have any real effect, but we have to actually run an experiment, which means we're going to have to take some people that have the disease and give them the drug, and some people that have the disease and give them something different, okay? Now, is it really ethical to actually be taking patients who have a disease that needs to be cured desperately and actually give them something that we know is not going to help them, okay, just because we have to have experimental control groups to figure out if the drug works? So that's something, if you were in that position, you probably wouldn't want to get the placebo, right? You would probably want to get something that you think is actually going to help you, okay? It could also be a situation where maybe the drug isn't as safe as we think it is, okay? And you don't necessarily always know that until you do very detailed studies. So one way to test some of these sorts of things first, before we actually give the drug to a person, is to treat animals with the drug, okay? And to try to make sure that the safety is what we think it is before we actually take it to the human population, okay? So these are all really important issues, and it's a delicate balance. Certainly, neuroscience, any scientist really is always careful to balance the cost and benefit of using living organisms to do experiments, and they do try to replace animal studies with any other type of study that we can mimic a living system without actually living or using a living system. So these are all things that are thought about continuously by people who do research and are factored into the equation when we're designing experiments ultimately that are going to help us understand brain function. Right? So that's just sort of some background stuff. Little trip into history is always fun. So I hope that you took at least a few important points from this. And what I would like you to do is watch this video here, because this video is actually one that I watched and I enjoyed it a lot. So this series called Cool Jobs, and they had one on the neuroscientists, and it actually, for people who have the question, like many people had the question, like I had the question initially, is what do you actually do? What is this job actually about? Okay, so you can watch this video. The guy is pretty neat, and he does a couple of experiments on the stage and kind of makes you think about the different questions that neuroscientists deal with. Okay, so at this point, I'm going to stop with this lesson. You can go ahead and take this quiz, see how you did. I always recommend that you take notes while you're following these videos, because I say a lot of things that aren't going to be on the slide, but all this information is important. So we'll see how you did, and I will see you next time for the next lesson.